first want to thank Keith and Susan for giving me this chance to participate in this convention. I really wish we could do it in person, because one of the things that I really value is learning from patients, as well as interacting with the other scientists. But I want to have this be kind of a discussion. I mean, I will have slides and I'll talk to you about some information I want to share, but I also want you to feel free to raise your hand, I guess is the way they're doing it, during the talk, and I'll pause at different points so that you have a chance to ask some questions about what I've covered so far. And that way you don't just have to listen to me drone on for the whole 45 minutes or whatever. A little bit about me to start with. So I'm very honored to be a member of the scientific board for the CMTRF. I've been studying Schwann cell biology for many years. I'm a basic researcher. I'm also now part of a team that is doing a small molecule screen for CMT. We're focusing on CMT1A. We've done a huge screening of 2 billion compounds and are now starting to narrow it down and do further analysis. We also have a project studying what the problem is with the Schwann cells that have too much PMP22. We have some exciting data along those lines, but we're still working on that. And then on a personal level, I have a nephew who has CMT, and that's been very motivating and very eye-opening for me as a scientist. So let me go ahead and share my screen. Okay, can you all see that? I hope somebody can answer that.
Yeah, we can see it. If you want to stick it in presenter mode, perfect. Yep.
Okay, great. Thank you, Susan. So I'm going to start with a little history and just a little basic biology. If you'll forgive me, I'm sure many of you know some of this history in biology, but for those who don't, I think it's worth covering a little bit of it. So first, just who are these people that the disease is named after? So Jean-Martin Charcot is basically considered the father of neurology. He lived in the late 1800s, and he not only described CMT, but he described a wide variety of other neurological conditions, including things like multiple sclerosis. He made key discoveries for Parkinson's and other disease. So he was a very famous French neurologist. And he had a student working with him, a resident, Pierre Marie. And so they worked together in identifying what is now known as CMT. At the same time, there was a British neurologist, Henry Tooth, who described the disease as well. And so in naming the disease, all three get some credit for that. So let me just talk a little bit about some of the basic biology here, so that you can understand better about what's going on and why we're targeting different things in the approaches to therapies. So many of you have heard about myelin as being compared to insulation around an electric wire. I really don't like that analogy. I understand it, but I don't think that gives an adequate view of all the things that myelin does. And I'll have more to say about that in just a minute. So first, let's just define a few things. So CMT, especially CMT1A, is a disease of the peripheral nervous system. So your brain and spinal cord are the central nervous system, but there's a lot of other nerves and portions of the nervous system that are outside the brain and spinal cord. For example, your sensory nervous system that gives you a sense of touch and pain, that's peripheral. Your autonomic system, so like if you're speaking in front of a bunch of people and your heart's beating a little fast, your hands are kind of sweaty, that's your autonomic nervous system, the sympathetic nervous system. So those are the areas of the peripheral nervous system. And then I want to make one sort of little exception here for CMT. So motor neurons are actually the cell body, the main part of the cell is in the spinal cord, but it has projections that go out into the periphery. And those are myelinated by Schwann cells. And that also is a type of cell affected by CMT. So when we talk about neurons, this is a little diagram of a neuron. You see this big purple area here, that's the cell body. That's kind of the main part of the cell. That's where DNA is transcribed. That's where a lot of the main functions are. And then neurons have these long processes or these long fibers that are called axons. And they go all the way out to the terminals. So for example, a motor neuron up here in my spinal cord is going to project all the way down to my foot. So that axon is about 1 meter long. So that's a pretty long distance. And so those Schwann cells that wrap around that axon are really important for that nerve to function. So just keep in mind when we talk about axon, that's this long nerve fiber. And then the cell body, the soma, that's where like most of the functions of the cell are. And obviously there has to be a lot of communication up and down that long passage. So what we best know myelin for doing is increasing electrical conduction. So neurons send signals electrically from one end to the other. So for example, a motor neuron is going to send an action potential or an electric signal from the spinal cord all the way down to the muscle and allow your muscles to move. So for the motor neurons, it's important those are myelinated. And so this little diagram up here with the myelinated nerve, you can see that it's myelinated by single Schwann cells all along. So one Schwann cell will wrap around that axon, and you'll have multiple Schwann cells to create the full myelin sheath. And then there's little gaps in between those Schwann cells. And that's where the electric signal can jump from one place to the other. And that increases the speed a lot. And you can see the numbers here, up to even 100 meters per second. So it can go very fast. The one analogy I kind of like is dominoes. Instead of just one signal going along, you know, the domino kind of falls, makes the other ones fall. There's a picture here. I was going to include a video. If you want to have some fun, look up dominoes falling. Just Google it. It's amazing, some of the different videos that are out there. Anyway, so that's important, especially if you want that rapid signal, like if I'm trying to walk or if I'm, you know, sensing that I'm stepping on something like glass. I want to move my foot quickly, right? So those fibers are myelinated. But there's other fibers that are not. So sometimes you don't need that signal super fast. Like I cut my finger recently and I was notified that I cut my finger because it hurt when it happened, but I still have a little pain there. And that's just to remind me, oh yeah, you have a cut, be careful with that, make sure it's clean and don't hurt it any further. Well, that signal, the reminder doesn't need to be super fast. So that fiber, that axon, is actually not myelinated, but it is surrounded by Schwann cell. It just wraps around it, doesn't form that thick myelin layer though. And then these axons, or these long nerve fibers, get bundled together in the nerve. And so here's our little diagram of the neuron with the axon. And you can see that a bunch of these axons get grouped together in these bundles, and then the bundles get grouped together in the nerve. And so the bundles are kind of wrapped by this special cell type called the perineurium, and that keeps it protected and keeps those together. And then there's blood vessels that go through the nerve too. And of course, that's important for getting oxygen and nutrients and things like that. But those blood vessels form tightly together and they form a barrier, actually, kind of like the blood-brain barrier, but it's called the blood-nerve barrier. So things can't just get in and out real easily. Those axons are protected by the blood vessels being tightly sealed up. Unlike in other parts of the body where they're leaky and that's intentional, so you can exchange oxygen and nutrients and things very easily. In the nerve, it's a little harder. And that perineurium also then serves as a barrier as well. And again, it's to protect these axons. And then the nerve is surrounded by another layer called the epineurium, and that's again, just to protect the nerve itself and give it some flexibility. So I've already mentioned these sensory neurons, but just as a reminder, the sensory neurons are right along the spinal cord. So this is a little picture of the spinal cord if you were to section it. Oops. And then the sensory neurons are right next to it, kind of in between the vertebrae. And that's why you might have heard of sciatica, where you can get pressure on those sensory neurons from like a disc that is collapsing or slipped, that can cause stimulation of that sensory neuron and cause pain. And then I also mentioned how motor neurons are actually in the spinal cord, but they project out into the periphery and allow for muscles to be contracted. And so I mentioned that myelin does more than just this electrical insulation. And I want to demonstrate that with a couple of findings over time here that just illustrate how important these Schwann cells are. So there was this group in Germany where they did a genetic mutation that caused the Schwann cells all to die during development. And while the neurons that were born, the sensory neurons, they projected out to their target tissues, everything seemed fine. But shortly after those Schwann cells died, the neurons all died. And this is just a picture from that, where here you have your dorsal root ganglia, or where the sensory neurons are. And then you see many of them are dead without the Schwann cells. And the same thing happens if the Schwann cells have a dysfunction. So one of the speakers that we heard from in the scientific sessions yesterday, Jeff Milbrandt, they made a mutation in the Schwann cell. The Schwann cells were fine, but they just weren't functioning correctly. And as a result, they produced some toxic lipids or fats. And that caused the neurons to degenerate. And you can see that in this picture here. This is from a skin section. And here you see these little squiggly fibers going into the skin. But when they cause this mutation in the Schwann cell, you see a lot of these fibers degenerated and died away. And we've got a recent study that was just published, kind of the same thing, where there was a mutation we found in the Schwann cell. The Schwann cells are fine. They actually myelinate fine, but they're producing toxic metabolites that the neurons don't like, and the sensory neurons died. And then the other thing that we're discovering is there's a lot of communication between axons and Schwann cells all through life. Obviously, they're providing something that's necessary for those neurons to survive and to function normally. We don't really know what all that is. We know there's some energy that can be provided from the Schwann cell to the axon. We don't really understand that. There's lots of different kinds of signals that go back and forth, and even whole chunks of stuff that Schwann cells have for their own function can get into a neuron. Just as an example, some of you might know what a ribosome is. It actually makes proteins. People have shown that whole ribosomes can go from a Schwann cell into an axon. So there's a lot going on between the neurons and the Schwann cells. And then one important thing I want to mention is that Schwann cells actually promote regeneration. So that's very different than what we find in the central nervous system, where the myelin there it's different. It's made by these cells called oligodendrocytes, and the myelin inhibits regeneration. So here, the Schwann cells are really great because they promote regeneration. Okay, so let me just take a little pause here and see if people have some questions about some of the basic biology. I'll get a bit more into the diseases in a second. So how often is it seen that CMT1A patients have diagnosis of essential tremors? If so, how are the two related? My father and I both have CMT, I guess it's CMT. I've been genetically typed 1A along with several family members on my father's side family. My father had a deep brain stimulator installed to help with essential tremors, which it has. So I'm not a clinician, full disclosure. I'm a basic scientist. And I can say that the different symptoms exhibited by people with CMT, and especially CMT1A, are quite varied. It really depends on, like many things, the environment and other aspects of your genome. So, you know, the essential tremors, that could be caused because of the loss of myelin. Even in the mice, we see some shaking. But in that case, it makes me wonder if there might be more going on. And obviously, deep brain stimulation is going to be affecting the central nervous system, not the periphery. Is there any inflammation involved with the disease process? And is anti-inflammatory diet recommended for 1A? That's a good question. And actually something we've looked into recently as well. So yes, there is some inflammation with CMT1A. We do see some immune cells like macrophages getting into the nerves. This can also be seen in some of the mouse models. And again, I'm not a clinician, so I can't advise you about taking anti-inflammatories. But I will say that in terms of a diet, that an anti-inflammatory diet is generally a good idea anyway. I try to eat, you know, a fairly anti-inflammatory diet. And of course, now what that means is open to some interpretation, but I would say that if you are doing something that is helping suppress inflammation, it's going to be better for you overall. Another question here, are the spinal roots always enlarged in CMT1A? And does this often cause nerve compression? So this is something I've learned about in the last year or so. Another way to test for CMT is looking for nerve enlargement, which seems counterintuitive to me because we think about demyelination and loss of axons. The enlargement of the nerve, I wouldn't think would happen, but actually what does happen is these Schwann cells will kind of compensate for the loss of the axons and produce a lot of this matrix stuff. Kind of like collagen is part of that. So you get actually an increase in the size of the nerve. Again, probably not everybody, but in many of these patients. And so sometimes they can use even ultrasound to look at the nerve thickening. And does this cause nerve compression? I don't know. I would suspect that it probably depends on where it happens. But I think in general, the pain that can be associated with CMT happens due to things we don't fully understand. So whether that could be a contributor, I'm not sure. Lecithin supplement for 1A. Actually, that's a good question. So with that, let me go back to sharing my screen because I'm going to come to that actually and talk about lipid diets for CMT, especially CMT1A. Okay, so as you already know and have heard about here, there's a wide number of genes that can lead to CMT. And you know, I want to emphasize again that many people have not heard of CMT. Probably many of you didn't until you were diagnosed. I always ask graduate students and medical students if they've heard of it. Most of them haven't, the vast majority, but it's actually the most common hereditary neuropathy. So it affects a lot of people. And the most common form is CMT1A. You can see the forms broken down here in this little pie chart. And here's the duplication of PMP22, and that's the most common thing. And we now know there's 100 different genes that can be mutated and cause some form of Charcot-Marie-Tooth. So let me explain a little bit about what this gene duplication, what that means. So as you all know, or may recall, we all have 46 chromosomes, 23 from mom, 23 from dad. And the ones from mom and the ones from dad are copies. So everybody has 2 copies of every gene. The only exception shown down here is the X and Y chromosome. But for CMT1A, the chromosome we're interested in is number 17 here. And that's because the gene for peripheral myelin protein 22 is on this segment of that chromosome, and that gets duplicated. Now, most commonly, it's caused, or the duplication is something that is genetically inherited, but there are occasions when this will happen spontaneously. And this whole region, this large chunk here of the chromosome gets duplicated. And we now know that the key gene involved is this PMP22, peripheral myelin protein 22. Because even if you just like overexpress that gene in an animal, you'll see a lot of the symptoms that are very similar to what patients have. And there are other regions of DNA in this chunk that gets copied. And there is active research being done to figure out if some of those other pieces might be contributing. Because as I'll explain, things are a little more complicated in trying to understand what's going on. So I think a lot of this I've covered. There's a duplication that causes CMT1A. It affects people all over the world, all ages, all backgrounds. The symptoms, you inform us as researchers about the symptoms, but I just want to mention that often it's distal parts, because those long axons going all the way out to those muscles start to have problems sending the signal. And so the muscles aren't getting the signal to function. And so you get kind of muscle wasting in the fingers and toes. And that's where things usually start. So you'll see, even infants where they're having a little bit of trouble moving. People are often, when they're younger, a little clumsy. You might have some problems with the hands and the feet. And it is a progressive disease. So very, very slowly, it gets worse. What does the pathology look like? So again, here's our little neuron and the myelin. And if we cut that, cut it, and then now look at this section here where, sorry, this is very sensitive. The nerve is coming out at you. And so all those donuts that you see there, that's the myelinated axons. That's a healthy nerve. What you see over here is some of the pathology or the problems you see with nerves with CMT1A. So you can see there's these, what they call onion bulbs, where the myelin's kind of decompacted. It's like the Schwann cell is trying to form myelin, it's not doing a good job, and it keeps trying. And then it can also lead to these huge thickenings called temacula, which just means sausage. And then you also can see frequently axon loss, which is another thing that's bizarre. Like, I've already told you how important these Schwann cells are to the axons. What is happening? The Schwann cells are still there. They still could form myelin if everything was normal, but somehow they're causing the axons to degenerate. And we don't know how. We don't know why. So let's focus a little bit on peripheral myelin protein 22 and what it is. So this is a diagram here of all the amino acids in PMP22. It's in the membrane, or like I said, those Schwann cells wrap around the axon. Think of it like rolling up a poster. And so the Schwann cell is forming this membrane, this kind of like fatty material that goes around and around the axon. Okay, and in that fatty material is this protein PMP22. And then what it's doing there, we don't really know. we know it's necessary to get normal myelin. We know if it's either too much or too little, or if it's mutated. And I should have pointed out, all of those little orange colored amino acids, those are all the point mutations we know of in PMP22 that lead to Charcot-Marie-Tooth disease. We call that type 1E now. So one idea that's out there is that PMP22 is important for these connections between cells called junctions. So if we look at the axon here, this nerve fiber with the Schwann cell, and then we cut it so we can see the wraps of the myelin, you can see that there are these places where the cell, the Schwann cell either has to form a tight junction or it communicates with the axon and actually comes down tight right to it. So PMP22 seems to be involved in forming those, but we're not sure about that. All right, so why is too much PMP22 a problem? So again, maybe I'll just take a pause here. It looks like we have some other questions that have come up. So before I get into some of the details of the biochemistry or what the problem is, again, I'll come back to this lecithin question in just a minute. It is definitely worth something to consider. So another question here, is there anything that can be done to reduce the occurrence of fatty atrophy caused by CMT? I think that one of the things that is really helpful with CMT, and I've seen this with my nephew, and I'm sure many of you are engaged in some physical therapy, the more that you can keep those muscles working, at least to some extent, the better, because that'll prevent the deterioration of the muscles that is often seen. And then as the muscles deteriorate, you get fat deposition. So I would say physical therapy is a big factor. But you know, this is a progressive disease. It's not going to stop it. So I think what we need to do is find ways to cure it. And I'll say more about that in a minute. Why is there such wide variation in how CMT type 1A affects people? Yeah, some people have really mild symptoms. Other people have really extreme conditions. And really, we don't know. But again, as I mentioned earlier, I think we have to consider influences of the environment. Lots of things can affect the peripheral nervous system in the environment, and other genetic factors. So maybe, you know, somebody has a variety of different genes that are functioning a little atypically. Not that you would get a disease on its own, but if you have them combined all together, then you get a more severe form. But that's something that is being explored, and we really don't know. Let's see. So can you explain why patients experience a progression of the disease when going through hormonal changes like adolescence or menopause? That's an interesting question. And I think there's a lot of possibilities of hormones influencing disease. I don't know if I want to say progression, but at least the acute symptomology. For example, one of the clinical trials that was considered for CMT1A was a progesterone antagonist, but this had such severe side effects that was stopped. So that never progressed into people. But obviously, progesterone does have some effect that's really not fully understood. I have heard anecdotally from patients that sometimes when people are under stress, they have more exacerbated symptoms. So stress increases cortisol, that's another hormone. So I think there's a lot we don't know. And I do think that, you know, hormonal changes are likely to have some influence on the progression of the disease, but at this point in ways that we really don't know. Do I see any concern with the COVID vaccine with it being RNA-based, how CMT affects RNA? No, I don't think there's any problem. I haven't run across any literature indicating that the COVID vaccine would have any effect. Intellectually, it doesn't seem like a likely thing anyway, because that RNA is being targeted to actually produce the protein that your body reacts to fight off COVID. The CMT antisense, like if you were taking some of those clinical trials with the antisense, that targets a different RNA. RNAs are very different, just like proteins. So I don't think there would be any likely problem there. Can high blood cholesterol be caused by CMT1A due to the nerve breakdown? I doubt it. I have not seen anything on that. I think that that's unlikely. We do know PMP22 has an influence on lipid metabolism, and I'm going to come to that in a few minutes. But that's in the Schwann cell specifically, not globally. Globally, your whole body, the metabolism of cholesterol and fat is controlled by a whole bunch of different systems. So I don't think that's likely to have an effect. Is the experience with my nephew, does wearing AFOs help, or do they lead to faster muscle deterioration? So my nephew actually has a form of CMT type 2, and he's actually very, very severe. So his situation is different than CMT1A. I do know people that have CMT type 1A, but I think I really can't answer that question.
I wouldn't mind answering that question anecdotally, if you don't mind, Dr. Carter.
Yeah, please.
I wear leg braces and I have 1A. I'm 50 years old. I wear my leg braces because they keep me from falling and they give me much longer endurance, and they're, you know, my muscles are very happy when I'm wearing them. I also am at a position where I can choose to not wear them. And when I know I'm going to be safe, I choose to not wear them, also because I want to keep working out my legs. So my leg braces, and this is me anecdotally, I'm not a doctor, I'm not giving any medical advice here, but you asked anecdotally, so I'm going to answer anecdotally. You know, my experience is my leg braces allow me to do things that I wouldn't be able to do if I didn't have them, like walking and looking up at the trees or looking up at the sky, and going on some pretty rigorous hikes. But I can also choose to not wear my leg braces if I feel like working out my legs. So that's a balance I'm always looking at and thinking about. It's a fair question. As a foundation, we don't really offer that kind of advice. And I can tell you people who do, but that's my anecdotal answer.
Thank you, Susan. And
yeah.
There's a question about sexual function. Again, I can't answer that. I don't have CMT myself. I can say that sexual function is complex, and the systems that are involved there are many types of peripheral nervous system, like sympathetic especially. The sympathetic and parasympathetic regulate erectile function, for example, but that is not affected by CMT1A. But you know, maybe Susan can comment on that. I really don't know about some of the other sensory aspects of that.
So I've been asked this question a lot, and I have passed this question along to the clinicians, especially the people like Mario and David, who are seeing CMT patients regularly. And it's not a question that they're asking, and it might be for that reason, Dr. Carter, that it's not the same types of peripheral nerves that are involved. But I can say that they're not tracking that information, or the last time I asked, they weren't tracking. So there's no clinical evidence of it, but they're not asking the question.
I definitely think it would be something worth bringing up with clinicians, you know, people that are seeing lots of CMT1A patients, to find out if there's any effects there as well. Because again, it's mostly not the nerves affected by CMT1A, but sensory neurons can be affected, and that would, cause some reduction in sensation. Okay, so let me jump back to screen sharing and tell you a little bit more about the problems of having too much BMP22.
And let me jump in for a second and say we have about 10 minutes left. So if you want to get through your slides, and then I love the questions that you all are asking using this function. If anyone wants to raise their hand and ask a live question, you can do that as well. When Dr. Carter's done with this session or segment, I'll call on people's hands. So feel free to do that if you'd like.
Okay, great, thanks. Okay, so, kind of speed up a little, get through some of this. One idea is too much BMP22 causes these aggregates of protein in cells, kind of like the aggregates you think of in things like Alzheimer's. And one idea is that those aggregates plug up the trash can in the cell. So the cell normally has garbage it cleans out, and that's called a proteasome. But that doesn't really seem to fit because we don't see aggregates in CMT1A patient samples. And even the amount of protein that we see doesn't really correlate with disease. So there was a study done looking at BMP22 levels in skin biopsies, and I won't go through the graph, but basically, just because you have more protein does not mean you have worse disease. And conversely, if you had protein that was even a little below what most people have, you could have severe disease. So the relationship with the excess protein is a little unclear. So this gets to the question about lecithin and fats, basically. So as I mentioned, the Schwann cell wraps around the axon, and that wrapping is very rich in lipids, or fats and cholesterol, things like that. And so there's been some recent evidence that BMP22 can affect how much cholesterol is in those membranes. And when people analyze the nerves from CMT1A patients and mice, they found that there were some specific lipids or fat-like molecules that were changed, and they were reduced. And those were called what we call phospholipids. So lecithin is actually one of the things that is a phospholipid. It's phosphatidylcholine. So these researchers took rats that are a CMT1A model, and they fed them diets that had an enrichment of these phospholipids, specifically phosphatidylcholine and phosphatidylethanolamin. And they saw a significant improvement in the myelination, how many axons were myelinated. They also saw improvements in grip strength, and there was a modest effect on nerve conduction velocity. Of course, one of the big open questions, though, is how does BMP22 do this? How does it regulate lipid metabolism? So I wouldn't run out there and start eating a ton of phospholipids because we really don't know, number one, if this translates into humans. There's been a lot of cancers that have been cured in mice that didn't translate into humans. So we always have to be very cautious in interpreting any results. Could there be something here? Perhaps, but we really don't know yet. The other thing that BMP22 in excess seems to somehow change is in Schwann cells, there are some pores in the cell that let calcium in. And that's important, and every cell needs some calcium. But if you have too much BMP22, for some reason, it seems like you get too much of this pore allowing calcium in, and that causes problems inside the Schwann cell. Now, there's pretty limited evidence for this, but nevertheless, these researchers have gone forward with some clinical trials to try and block those calcium pores. And I want to emphasize that, we've talked about some of the problems here, but keep in mind that the exciting thing is that the peripheral nervous system can regenerate. So if you cut a nerve the distal part of the axon deteriorates and the Schwann cells actually become more immature. And those immature Schwann cells actually stimulate axon growth. And then once the axons grow, the Schwann cells will remyelinate. Now, as we get older, they're less efficient at doing that. But the point is, you can regenerate the peripheral nervous system in a substantial amount, unlike the CNS, the brain and spinal cord. So that's really promising. And in fact, there was this group shown here from Germany where they created a mutant mouse that had excess PMP22, and they had a way they could shut off the PMP22. So when it was too high, you can see you get this thin myelin, the donuts are not there, and they had all these problems. But then they shut it off when it was an adult. And they looked 1 week later and they saw remarkable recovery of the myelin. And they started to see recovery of the axons. So that means if we can find a way to shut off the excess genes, you can actually cure the disease and then not even just stop it, which is good. We all want this just to stop progression. But in theory, you could actually restore it, which is huge, right? And I won't go through all the clinical trials. There's a ton of clinical trials out there going on, which Keith mentioned already and you've heard about. And then I just wanted to point out, you can go to the CMTRF website and get more information about getting involved with that. There are limitations to clinical trials. I already mentioned we have a variety of different models. I'm not going to go through everything again for time's sake, but we might be able to fix this in a mouse. We just need to make sure that translates into a human. Getting things across that blood-nerve barrier can be a challenge. You've heard a little bit about that. It's like getting things across the blood-brain barrier. It's difficult, and you might not want things to get across very easily. Measuring outcomes is a huge challenge. For something that progresses very slowly, pharmaceutical companies don't have a ton of patience when they're looking at whether a drug works. They don't want to wait years and years to see if a drug works. They want to get the information as fast as possible so they can stop wasting money if it doesn't work. So there's a lot of effort being put forward to look at markers of progression. But again, this last slide here, I just want to end with the idea that this can be fixed. If we actually find a way to correct the problem, in theory, you could get regeneration and restoration of the myelin, thanks to the peripheral nervous system. Okay, and I'll stop there and be happy to take any other questions.
I also want to point out that the folks from DTX Pharma are here in the room, so we can take questions. We can ask them some questions too. And Denise or Brian or Artie, if you want to answer those questions, we can raise your hand. Dr. Carter, if you want to answer this one from Matt, and then we've got one hand raised.
Okay. Is there anything to be done to reduce the occurrence of fatty atrophy caused by CMT? I'm not sure if you mean by fatty atrophy the accumulation of fat. I think I kind of already talked about that with the muscle being replaced by fat if you're not using it very much. I guess that's what you're talking about. If not, you can follow up with more question.
Yeah, that's how I read it. Go ahead, Angela.
Hey, so I did sit through yesterday as I work for a CMDO. So great stuff happening and enjoyed the presentation today. Looking at where you're talking about the demyelation and actually being able to regenerate that, where you have the demyelation happen and then you have the atrophy of the nerves themselves. How does it impact, and is it believed or thought that at a certain state, because with the degeneration of the nerves, you also have the atrophy of the muscles, just trying to understand the sequence of events, how it might be able to help muscle mass regenerate, or is there any expectation for that with any of the studies that are occurring?
I think with the studies that are occurring, and maybe the DTX people could speak more to this, but they just want to see the progression stop or slow down. Actually curing is a wonderful goal, but that's a much more challenging thing. And I would say that when I talk about the regeneration and restoration of myelin and everything, that's going to take time. It's going to take considerable time. And then to build back up that muscle, it'd be the same thing if you've like ever, broken an arm or something and that muscle atrophies, it takes time to build that back up. So the recovery, even if everything goes great and you regenerate and remyelinate it, it's going to take quite a bit of time to restore function back to what would be considered typical.
Thank you.
Thank you. So Harold asks, do DTX folks think participation in the FARNEX trial will exclude someone from your trial? I don't know if you want to raise your hand, one of you, and I'll let you speak. I'll give you guys a minute to do that. And in the meantime, Michelle asks, Bruce mentioned that the level of protein expression does not correlate with the severity of symptoms. Why do we think that reducing the protein will then solve the problem and cure the disease?
Yeah, great question. So the thing is, we're not actually targeting, at least not that I know of, there isn't any targeting of the protein. They're targeting the gene or the messenger RNA. So don't forget, you go from gene to RNA to protein. And so maybe there's something about the RNA levels that we need to reduce those, and that then will alleviate the problem. Honestly, we don't really know, but all I can tell you is that the amounts of protein do not reflect the neuropathy that people experience. So one idea actually that John Sferrin has and June Lee is that you have these fluctuations in protein levels, and that's the problem. It's not that you just have too much and it stays too high. It's that there is actually fluctuations because that whole region is duplicated. And so the gene regulation is all messed up. So you get these fluctuations, and maybe that's the problem, but we really don't know.